Welcome to our very first lecture on insect vectors and diseases. Today, we are going to explore a fascinating but dangerous condition known as Chagas disease. Imagine a disease that can hide in your body for decades without a single symptom, only to strike when you least expect it. By the end of this video, you will understand exactly how this silent killer operates and how we use mathematics to stop it. To understand Chagas disease, we must first meet its vector. Imagine sleeping peacefully, completely unaware that a small insect has crawled onto your face. This is the kissing bug, named because it prefers to bite near the lips or eyes. But here is the crucial part. The bite itself does not transmit the disease. Instead, the bug defecates while feeding. When you unconsciously scratch the itchy bite in your sleep, you accidentally rub the infected feces directly into the wound or your eyes. This sneaky nighttime invasion is the starting point of the infection. Now that we know how the bug attacks, let us look at what it leaves behind. Chagas disease is scientifically known as American trypanosomiasis. The true culprit is a microscopic protozoan parasite called Trypanosoma cruzi. The kissing bug, which belongs to the insect order Hemiptera, acts merely as a delivery vehicle. Once this parasite enters the human bloodstream through the scratched wound, it does not just float around. It actively invades your host cells, hijacks them, and multiplies rapidly, preparing to spread throughout your entire body. This brings us to a very important biological distinction. You are probably familiar with mosquitoes, which inject pathogens directly into your bloodstream through their saliva when they bite. The kissing bug operates entirely differently. Because it transmits the Trypanosoma cruzi parasite through its feces rather than its mouth parts, scientists call this a stercorarian transmission. Understanding this unique fecal transmission route is vital for zoologists and public health officials because it changes how we approach hygiene, prevention, and vector control in affected regions. So, what happens to the human body once infected? Chagas disease acts like a slow-ticking time bomb, divided into two distinct phases. The initial acute phase occurs right after infection. Surprisingly, it is often very mild or completely asymptomatic, meaning the patient has no idea they are sick. However, the parasites are silently spreading to vital organs. The disease then enters a chronic phase, where it can hide undetected for decades. Years later, it can suddenly manifest as severe and often fatal cardiac failure or massive digestive blockages. Let us visualize everything we have discussed so far. On the left side of this diagram, you can see the kissing bug taking a blood meal from a sleeping human. Notice the small deposit of feces left near the bite. The arrow labeled scratching shows how the host accidentally introduces the Trypanosoma cruzi parasites, shown in the magnified circle, into their own bloodstream. Following the arrows to the right, we see the human silhouette. The heart and digestive tract are highlighted in red. This represents the chronic phase, showing exactly where the parasites settle to cause severe, long-term damage if the infection is left untreated. Seeing the severe damage this disease causes, we must ask, why is studying this so critical? Historically, Chagas disease was endemic only to rural Latin America, affecting millions of vulnerable people living in poor housing conditions. However, with increased global travel and human migration, it is no longer confined to one region. It is now a global health challenge. By understanding the vector, we can implement life-saving interventions. These include improving rural housing to keep the bugs out, screening blood donations to stop human-to-human transmission, and applying targeted insecticides to eliminate the bugs entirely. To effectively deploy those interventions, we need more than just biology. We need mathematics. Epidemiologists use mathematical models to understand exactly how quickly Chagas disease can spread through a community. The most important concept here is vectorial capacity. Think of vectorial capacity as a measurement of transmission potential. It calculates the average number of potentially infective bites that will eventually be delivered by all the bugs feeding on a single infected person in one day. By quantifying this risk, public health officials can predict outbreaks and mathematically prove which control measures will be the most effective. Now that we understand what vectorial capacity is, let us look at the formula used to calculate it. C equals m times a squared times p to the power of n, all divided by the negative natural logarithm of p. Let us break down these symbols. C is the vectorial capacity. The variable m is the density of bugs per person. The variable a is the biting rate, or how often a bug bites a human each day. We square the biting rate because the bug must bite twice, once to get infected and later to transmit it. The variable p is the daily survival rate of the bug, and n is the incubation period, which is the time the parasite needs to develop inside the bug. Finally, the denominator represents the daily death rate of the bugs. If bug density or biting rate increases, the disease spreads much faster. Let us see this formula in action with a real-world example. Imagine a rural village where health workers find 5 bugs per person. Each bug bites once every 4 days, making the biting rate 0.25. The daily survival rate is 0.90, and the parasite takes 15 days to incubate. We want to find the baseline vectorial capacity. First, we calculate the raw biting pressure, m times a squared, which gives us 0.3125. Next, we find the probability a bug survives the 15-day incubation by calculating 0.90 to the power of 15, yielding about 20.59%. Then, we find the daily mortality rate using the negative natural log of 0.90, which is 0.1054. Finally, we multiply the numerator terms and divide by the mortality rate. The result is approximately 0.61. This means every infected person generates about 0.61 new infectious bites per day, indicating a high risk of ongoing transmission. Since a vectorial capacity of 0.61 is dangerously high for a chronic disease, the health department must intervene immediately. One of the most effective strategies is indoor residual spraying. This involves coating the inside walls of houses with a safe, long-lasting insecticide. The goal here is not necessarily to kill every single bug instantly on contact. Instead, the insecticide targets the adult vector's daily survival rate. Because the parasite requires a long 15-day incubation period inside the bug, reducing the bug's lifespan means it will die before it ever becomes infectious. As we will see mathematically, even a modest decrease in daily survival can completely collapse the disease transmission cycle. Let us calculate the impact of this spraying intervention. The insecticide reduces the daily survival rate from 0.90 down to 0.70. All other variables remain exactly the same. First, we recalculate the probability of a bug surviving the 15-day incubation. 0.70 to the power of 15 drops drastically to 0.0047. This means less than half of 1% of bugs now live long enough to become infectious. Next, the new daily mortality rate increases to 0.3567, meaning about 36% of the bugs die each day. When we plug these new numbers into our formula, the vectorial capacity plummets to a mere zero point 0.004. Physically, this means transmission has been almost completely halted. The bugs are dying long before they can pass on the parasite. We have covered a lot of ground today, so let us review the most important ideas. First, Chagas disease is a dangerous infection caused by the Trypanosoma cruzi parasite, uniquely transmitted through the infected feces of the kissing bug. Second, because the disease can remain hidden in the human body for decades before causing fatal heart or digestive damage, preventing transmission is our most vital tool. Finally, by using mathematical models like vectorial capacity, we can design highly effective interventions. We proved that strategies like indoor residual spraying, which reduce the bug's survival rate, can completely collapse the transmission cycle, ultimately protecting vulnerable communities from this silent killer. Great job following along with both the biology and the math.